Hello everyone, welcome back to my channel. In this video, I am going to talk about the importance of lipaemia, haemolysis and ICTERUS and its possible interferences in clinical pathological results. Lipaemia is otherwise called as lactulescence, which is caused by increased amount of triglycerides. Triglycerides are present as chylomicrons or high concentration of low density lipoproteins. Lipaemia is usually a postprandial artifact where the blood is collected from a patient who is not fasted. Lipaemia can be avoided by collecting blood from a fasted animal. A minimum of 12-hour fast is recommended to avoid lipaemia. If the lipaemia is present in fasted animal, then it indicates the underlying condition, which can be pancreatitis, hepatic lipidosis, diabetes mellitus, and some kind of neoplasia. There are three different mechanisms involved in interferences with analytes. First mechanism is light scattering, second mechanism is volume displacement, third mechanism is hemolysis. In light scattering, there is falsely increased absorbance of some analytes, for example total bilirubin. Hence, lipaemia can falsely elevate total bilirubin levels in the serum. In the mechanism of volume displacement or solvent exclusion, there is false decrease in some analytes. For example, electrolytes, mostly sodium and chloride are affected. Sometimes potassium can also be affected. Hemolysis is actually triggered by the presence of lipaemia in the serum. So the free hemoglobin in the serum can interfere in the absorbance of various analytes. Lipaemia can also affect various hematological parameters. Hemoglobin and hemoglobin related indices are falsely increased due to presence of lipaemia. So this will manifest as high MCH and high MCHC in the report. Lipemic serum can falsely elevate total platelet count because large lipid molecules can be counted as a platelet in CBC analyzer. Presence of lipids can falsely elevate total protein if you are measuring them with refractometry because lipids can refract light. There are ways to minimize artifactual changes due to lipemia in laboratory samples. This is usually done by refrigerating the lipemic serum overnight, followed by high speed centrifugation. By doing that, you will be able to separate the creamy layer which is rich in triglycerides and chylomicrons at the top, leaving the clear serum in the bottom. So for analysis, you can use the clearer serum. Coming to the interferences caused by in vitro hemolysis. Hemolysis interferes in hematological testing by two different mechanisms. One is lysis of RBC itself and the second one is light scattering. The hematocrit, PCV and RBC count will be falsely reduced with in vitro hemolysis. Hemoglobin related indices such as MCH and MCHC are falsely elevated due to hemolysis. If you are measuring total protein by refractometer in a hemolyzed sample, the line that indicates the specific gravity and total solid line is severely blurred because of free hemoglobin in the plasma. Platelet count can also be falsely elevated in cases which has intravascular hemolysis or artifactual hemolysis where ghost RBCs are mistaken for platelets while processing in CBC analyzer. Coming to the interferences caused by hemolysis in biochemical analytes, there are 5 different mechanisms involved in it. First mechanism is increased absorbance. Presence of free hemoglobin in the serum can alter the absorbance of other analytes in the serum. Second mechanism is inhibition of reactions. Free hemoglobin or released hemoglobin in the serum can directly inhibit certain reactions, which can alter the concentration of those particular analytes in the serum. The third mechanism is release of intracellular constituents that can be directly measured. When RBC ruptures, the contents are released in the serum. For example, potassium is an intracellular ion. So potassium can be elevated in hemolytic conditions. Enzymes such as AST, LDH, then magnesium, sometimes iron can also be falsely elevated. Phosphates are also falsely elevated in hemolyzed samples. The fourth mechanism is release of membrane or intracellular constituents that participate in certain reactions. For example, adenylate kinase or glucose 6 phosphate are known to increase the concentration of creatinine kinase in serum. Fifth mechanism is water release. So the ruptured RBCs contain water. So those water can dilute some constituents that are present in the serum already. So here I have an example of hemolyzed serum. This is a normal serum. This serum is mildly hemolyzed. And this serum is extremely hemolyzed. According to the degree of hemolysis, the changes in serum analytes will vary. Hemolyzed serum can also interfere in electrophoresis. In hemolyzed serum, during electrophoresis, the hemoglobin can travel to the beta region in the tracing, causing false elevation in beta globulin. In the same region, hemoglobin haptoglobulin complex can also migrate to that particular area. In severely hemolyzed sample, a tall monoclonal like graph with broad base is commonly seen in electrophoresis, which can be commonly misunderstood with monoclonal immunoglobulin spike. Coming to the icteric serum, it does not interfere with most of the hematological parameters, but it do interferes with some biochemical parameters. High bilirubin levels can falsely reduce the concentration of creatinine and total protein level in the serum. As a quick summary, lipaemia can falsely decrease sodium, chloride, bicarbonate, LDH. Lipaemia can falsely elevate magnesium and total ion binding capacity. Hemolysis can falsely elevate potassium, AST, iron, phosphate, LDH, magnesium, creatinine kinase, etc. Hemolysis can falsely decrease amylase, GGT and ALP. ICTERUS can falsely decrease creatinine, cholesterol and GGT. Thank you so much for watching this video. For more such videos, make sure you subscribe to my channel. I'll see you all in another video. Till then, take care and bye.